Welcome everyone to the Northern Vancouver Island Marine Mammal Science short evening. My name is Caitlin Birdsall. I am with the Marine Education and Research Society, and I am going to be the moderator of tonight. We're going to get started right on time, and hopefully people will filter in as well, because we have sort of a packed agenda of many presentations about research and conservation projects that are happening around Northern Vancouver Island this year. We at MERS are co-hosting tonight's evening with our friends at the North Island Marine Mammal Stewardship Association. And so we have Stephanie and Janine who will be talking in just a moment about NIMMSA and welcoming us all as well. I do want to mention that this is the second of two sort of pre-season events that we've hosted. The first we did host with our colleagues at Orca Lab as well. It was a community meeting held in Port McNeill on May 24th. And so many thanks to those of you that came out to that. It was a really fun and really inspiring night as well. We're going to talk in just a moment about things like Q&As and everything like that. But before we dive into the meat of this evening, I did really want to recognize that I am coming to you all tonight from the territory of the Liquidoc people, and that includes the Wiwaikai, Wiwaikum, and Quika people here in Campbell River. And that we respectfully acknowledge that the areas that we're talking about tonight and the areas in which we all work are the territories of multiple coastal First Nations who are sort of the original knowledge keepers and stewards of these waters and lands since time immemorial, and wanting to really put that forth early on tonight. So for now, I'm going to pass it over to Janine with the North Island Marine Mammal Stewardship Association so she can welcome us and give us some words about NIMMSA and tonight.
Thanks, Caitlin. Hi, everyone. My name is Janine Sutherland. I'm the advancement manager for NIMMSA. I actually just started in this role a couple of months ago, so I'm pretty new on the scene here, and I work with Stephanie. Steph, do you want to give a little wave there? So just wanted to say a big thank you for this evening. It's really so fantastic to have this forum, this opportunity to learn about research projects on the North Island. For those of you that aren't familiar with NIMMSA, we conduct conservation and business activities aimed at benefiting marine mammals, the economy, and the marine environment off Northern Vancouver Island. We have roughly 30 operators as part of our membership from a variety, representing a variety of different businesses such as whale watching, kayaking, private charters, lodges, and then we also have about a half dozen nonprofits that are members as well too, and MERS being one of them. And I'll chat just very briefly here a little bit about our conservation fund. If you want to just advance the next slide there. Yeah, so one of the cool things that NIMMSA does is we have a conservation fund aimed, we basically invite charities, nonprofits, research and academic institutions, Indigenous governments, coalitions to apply for funding for conservation activities on the North Island. And in 2022, we had 4 successful recipients. We had Bay Cytology do a project on multi-population killer whale sparring study. We had University of Alberta do a project on the population biology of spot prawns in Northeast Vancouver Island, Broughton Archipelago, and mainland inlets. And we had MERS, development of an online acoustic disturbance resource to support recreational boater education, and Salmon Coast collaborative kelp forest monitoring to support effective kelp conservation and restoration in the Broughton Archipelago. And then this year we were super excited because, well, we actually had request for about $92,000 worth of funding. We were able to fund about 38K worth of projects, but we've got some really exciting projects on the go. So Bay Cytology is doing an open access database for big killer whales. CETUS is doing Be Whale Wise for paddlers initiative. And then I always struggle when I say this, but I'm going to try my best here. Nuk Hawakmish First Nations is embarking on a project to protect beaches, promoting community values, and enhancing positive ecotourism in the Broughton Archipelago. And then MERS exploring the feasibility of a slowdown zone for large vessel traffic in Blackfish Sound and Black Knee Pass. And then Salmon Coast, understanding the health and migration of juvenile Chinook salmon. So we've got some really great projects, and basically the way that this fund works is all of our members contribute $1 per day per guest to go towards this conservation fund. So we're pretty excited about that. So without further ado, I don't want to talk too much. I know I've got my time limit here, but thank you so much for having me here this evening, and I'm really looking forward to the presentations.
Sorry, just I'm muting myself really quickly. Wonderful. Thank you so much, Janine, and really grateful to have NIMMSA as a co-host tonight, and also such a supporter of research and conservation projects on the North Island. Just really quickly, I wanted to kind of go over tonight's goal. And really, we're all gathered here tonight to sort of learn and share about the different research and conservation projects that are happening in the area that we all work. And this is really in hope to make sure that we're building community, that we are informed about what one another are doing, and so that we can work together respectfully as well throughout this season and into the future as well. We really also want to make sure that this is an opportunity to be able to recognize one another while we're on the water, and to know who to direct questions to if they come up throughout the season. So as I mentioned, this is the second of two sort of pre-season events. And last year when we held our pre-season meeting, we really heard that there's two different needs here: to build community, and that's what sort of our May meeting was about, to all get together, but then also to be informed. And so that's what we're trying to do tonight as well. So as I mentioned, we do have a jam-packed agenda. I do want to mention that this event is being recorded in case there are folks who are not able to join us tonight, but would like to see the presentations. We have 9 panelists who will be giving quick talks, giving overviews of their projects for 2023. Each of them will have 5 minutes to speak, and then another 3 minutes for questions. And I'll talk in just a moment about how to ask questions. I do want to let our panelists know that I will be letting you know. I'll be piping up when your 5 minutes are done, and I apologize in advance if I do have to interrupt anyone. For questions, for those of you that are with us today, we're going to ask you to put your questions into the Q&A, and you should be able to find that in the bottom panel of your Zoom tonight. So type them in as they come up. You don't have to wait till the end. Please use that as much as you need to. Marika, who is off, she can give a little wave. Marika is one of our MERS team. She's going to be monitoring the Q&A, and then she'll be sharing the questions after each presentation for the presenter to answer. If there are any sort of outstanding questions that we're not able to address in the time limit at that point, Stephanie with NIMMSA will be holding on to those, and she'll ask them at the end of all the presentations if we have extra time. If we really run out of time, we really want to make sure that we are answering those questions. So we'll hold on to them, we'll distribute them to the presenters, and then we'll email those answers. Because again, the goal here really is to be able to be informed and to learn about what one another is doing. So we want to make sure we're addressing those questions, but that we're also sticking within the time limit. So that's it for me. Our first presenter tonight is going to be our colleagues with Oceanwide Conservation Association. So I will pass it over to Brittany and Gary.
Thanks, Caitlin, and good evening, everyone. I'm Brittany Vasona Kelly, And I have my colleague Gary Sutton here with me. We're part of OceanWise's Whales Initiative, and we're going to talk about two of our projects that have field components along the North Island. So the first project is a drone-based study that is looking to assess the body condition of humpback whales. So this project has two main goals. The first being to use these aerial images to measure their conditions, so look at how fat these whales are and relate that to prey abundance, as well as their foraging areas and any changes annually. And the other goal of this project is part of a collaborative study with MERS. And so it's to use those images to look at rates of entanglement and ship strikes. And they'll talk a little bit more about that when it's their turn. As for field efforts in 2023, our kind of study area is from French Creek up to Johnson Strait. We're tentatively planning maybe a couple of days early August or early fall, but it's all opportunistic, and we'll be working around other people's schedules on when they're there. We don't want to put added pressure on the whales or to have any overlap. So we'll be working with everyone else on that. The vessel that we'll be using is OceanWise's research vessel. So as many of you have already known, the Scanna. So that is what you'll see when we're out and about. Our second project is a winter study project. And this is basically to address some of the gaps in the recovery strategies of many at-risk cetacean species. So helping to document their habitat use as well as their diet in winter. So our goal there is just to basically be out on the water opportunistically, collecting any sightings, as well as collecting prey samples from resident killer whales or humpback whales to provide to DFO as part of their ongoing prey study. We're also hoping to document what these species are doing with regards to the herring spawn. If they're using it, what areas are most important, and if there's any fisheries interactions. The other main goal is to collect eDNA samples. So as from killer whales and humpback whales. And as these animals dive, they will shed some skin cells, so you can collect water samples from their fluke print. So it's a non-invasive method, and you can look into genetics of individuals, sexes, and genetic diversity. So we're hoping to see the feasibility of this method, as well as how it works for modern population demographics. And I'll pass it to Gary to talk a bit about the fieldwork for the winter sampling project.
Thanks, Britt. Yeah, so our first winter field season went really well. It was a bit delayed due to some unforeseen issues, but regardless of that, we actually had a great field season. We were based for the most part in Cowichan Bay, operating around the Southern Gulf Islands and the lower Strait of Georgia, but it did bleed a little bit into the North Island when we were up Hornby Island, which we can plan to continue to do so. So we'll probably see you guys up there, at least some of you. I saw Wild Waterways Adventures a few times up there this past year. And moving into this next coming year, we're hoping to get a much earlier start. So starting in October, which will hopefully bode well for a lot more samples, especially for humpback whales. Next slide. So just a quick little update. So despite our late start, we still, like I said, had a great one. We had 4 southern resident killer whale encounters, 17 big killer whales encounters, including some really cool ones off Hornby Island of some sea lion killer whale interactions, which you can see from the bottom right photo there, are always fun to watch. And but probably the most notable, even though we only had 3 humpback whale encounters, the top right photo, we had a really cool one in Lambert Channel, where we had this young humpback whale feeding on spawning herring, which was, to our knowledge, the first time that's been documented. So it was really exciting to see that, and to see how this kind of story will unfold moving forward. And this is just a quick little summary of the samples we were able to get. So for southern residents, we were able to get 23 eDNA samples, which all came from JPOD, as well as 1 prey sample. Humpbacks, we only encountered 4 individuals, but we got all of them. We got 4 eDNA samples and 2 prey samples. And then for bigs, we got 37 eDNA samples from them. And that's it. So just a thanks to our funders and partners. And if you guys have any questions, let us know.
Perfectly on time. Good work, you guys. That's fantastic. Marika, it doesn't look like there are any questions for the OceanWise team. So yeah, thank you guys so much. Yeah, we look forward to seeing you on the water this summer. So next down the pipe, and if there are questions that come up for Brittany or Gary after the fact, you're welcome to still pop them in the Q&A and we'll address them at the end. So the next presenter we have is the folks from Salmon Coast. Caroline, are you there?
Hi, so sorry.
Wonderful.
All right. Hi, everybody. Sorry about that. I'll just go ahead and share my screen. All right. Can you guys see that all right?
Not the presentation, Caroline. We see your full screen. You're good now.
Okay. Okay, awesome. So yeah, my name is Caroline Glass and I am the station coordinator at Salmon Coast Field Station. And I'm going to talk a little bit about our project looking at kelp health and the repopulation of sea otters to the Broughton Archipelago area. So for those of you who don't know, Salmon Coast is a fully off-grid remote field station based in, again, the Broughton Archipelago on Guilford Island. And we conduct our research in the Musqueam Zawadanic waters, and our field station is located on Kwikwasutunu-Kakwamis unceded territory. So in 2021, last year, we started a project looking into the distribution, health, and significance of kelp beds within the Broughton Archipelago. The project is a collaboration between the Broughton Aquaculture Transition Initiative, the UVic Spectral Lab, and the Kelp Rescue Initiative. And it was started because local people around Alert Bay started reporting declines in the abundance of kelp in the area. So we have undertaken a couple of different aspects of the project, some investigating whether juvenile salmon are using kelp beds, conducting underwater surveys to identify key metrics of kelp health, and conducting shoreline distribution surveys to identify presence and absence of kelp within the Broughton Archipelago. And another component of the project that we are looking at is how sea otters interact with kelp, specifically bull kelp here. As many of you probably know, we have started seeing the repopulation of sea otters to the area. And it's now common to see rafts of, larger rafts of sea otters around Alert Bay, Malcolm Island, and Port McNeill. The sea otters play an important role in the maintenance of canopy-forming kelp beds as an apex predator of the marine nearshore environment. They can alter community dynamics through predation of sea urchins, which can destroy kelp beds if left in high numbers. Because of this relationship, and because the repopulation of sea otters happened, fairly recently, we are in a unique position to be able to really study the direct effects of the sea otters on kelp bed growth. There's a couple aspects to the project. So the first is working off of the work of other people such as Jackie to do a bit of distribution studies of the sea otters. So these are surveys where we send out a few viewers on our boat to look around the area, typically looking around 100 meters off the shoreline and just documenting where they're seeing the otters, how many otters there are, and trying to identify things like the makeup of the group, looking for pups and feeding behaviors. And we've done a few surveys which have kind of supported what has been found previously and anecdotally. They saw between 30 and 60 otters at places around Cormorant Channel, Pierce Passage, and Alert Bay. We also have been starting to get more reports, and we're here at Salmon Coast are seeing single otters within the Broughton Archipelago. So we're planning on expanding and doing some more surveying on this side of Blackfish Sound. And we are also adding a citizen science component to this project. So we'd love to pass this on to everyone here, anyone else you might know within the area. And that is getting folks who are in the area to use iNaturalist to report their findings of sea otters. So that way we can use that. There'll be so many more eyes on the water, especially over the summer. So getting folks to share that location will really improve our ability to study where the otters are and their impact on kelp. So I'll probably be reaching out to folks in the next couple of weeks to distribute this poster, and yeah, we'd love support on getting the word out there about this. The second aspect of the project is actually looking at the kelp beds themselves to see how the otters are affecting the size and growth rate of the kelp beds. And so we'll be doing this by looking at two different kinds of kelp beds, some where there are otters currently, and then others where, after doing some habitat suitability analysis, as well as talking to sea otter experts, where we think that the sea otters might be going. So the kelp bed surveys will involve looking at things like density of the kelp beds, size and area of the kelp beds using drone surveys, and then using underwater footage from ROVs and drop cams to get a sense of urchin presence, substrate, and understory kelp growth. And yeah, the idea is to do this over a time series, so doing it now while the sea otters are just moving in, and then after they have moved into the area to get a sense of what's changing within those kelp beds.
Hi, Caroline.
And we'll be doing that using the DRAT to measure the density of the bed. Oh, sorry.
Oh, no, you just cut out there for a minute. So
It's just a trick.
We're almost at the end.
Oh,
We're
sorry.
at the end of your 5 minutes too. Yeah.
Sorry. Okay, awesome. And that's just an example.
Sorry, Caroline, I think we've just lost you again. I can't hear her. Can anyone else hear her right now? No.
Negative.
No.
If Caroline can hear us, shutting off her video would work.
Yeah, if you want to shut off your video there and maybe wrap up really quickly. Oh, there we go. Moved you forward.
Here we go.
Okay.
All right. Yeah, well, thank you to everyone. And yeah, thank you to all our sponsors and NIMMSA. And thanks for having us here tonight.
Wonderful. Thank you, Caroline. I know that there are a couple of questions here. Just to remind everyone, we are using the Q&A, but Sharon, I did see your hand went up. And so Marika can quickly unmute you and you can ask that question. And then I think we have a question from Lance as well. Great.
Sharon, you should be unmuted.
That was a mistake. Sorry.
Oh, okay. She was just waving hi. Okay, Marika, do you want to read out Lance's question then?
Yeah. The question was to ask if the otters are all fairly habituated to boats or whether some are boat shy, as they tend to be on the Central Coast in Lance's experience.
Sure, just from my sort of anecdotal experience and our experience from doing the surveys, many of them are fairly habituated to the boats, and I've seen boats get quite close without any reaction from the otters, especially when they're in the larger rafts. But there are, when you see them more individually, I have seen them more wary of the boats. And also generally in rougher weather, I've found that they are a bit more boat shy. Awesome.
Fantastic. Thanks so much, Caroline. Very, really interesting. And we're really glad to have had you here today. We are perfectly on time. You guys are amazing, all of you. I'm going to pass it over to Strand Tucker with Fisheries and Oceans Canada to give us an update on some of the pinniped work happening locally.
Hey, assuming you can see my screen. Perfect. Great. Well, thank you so much for having me. Yeah, I'm currently the head of the pinniped research program at DFO in Nanaimo. It's part of a team, definitely a team effort for the work we do. I've just put everyone's name down here. So we have two projects happening up in the northern part of Vancouver Island, one related to harbor seal telemetry, the other pinniped diets in general. So just to give you a little background on why we're doing this. So basically the satellite deployments relate to improving our population assessment. So essentially, determining the abundance of seals is a two-step process. We obtain a census from aerial surveys. Our aerial surveys are undertaken during summer pupping season. And then we subsequently apply an upward correction factor based on the proportion of seals that are actually hauled out during our very specific survey window. And this correction factor is based on the behavior of seals derived from satellite telemetry. So we're essentially tracking the proportion of seals that are hauled out during our window. So prior to the work I'm subsequently going to show you, the last deployments to derive these correction factors were done on seals in the Strait of Georgia in the 1990s. So we had a tagging program between 2019 and 2021 where we were able to revisit these correction factors, and we had despite COVID and everything, we were able to get out and about, and we had deployments about 32 seals in the Strait of Georgia. This is just an image of our tags here. You notice this yellow float is foam float. We're actually able to recover these tags in the Strait of Georgia and get all the data that these tags log. So obviously we're also collecting, depending on the tag, additional ecological data on diving behavior and foraging distribution and so on. But the impetus is this correction factor derivation. So just to sum up here, what we observed from our deployment period, that almost 80% of the animals were hauled out during our survey window. And this was a dramatic change from those first deployments in the 1990s, where only 62% of the animals were observed to be hauled out. So any number of hypotheses there, potentially related to the large increase in or concentration of transient killer whales in the strait, or potentially high abundance of spawning herring biomass. But anyway, this has a large impact on our final population abundance estimates. However, in British Columbia, there have never been any deployments on seals outside of the Salish Sea. So this summer, we started to attempt to rectify that situation. Just anecdotally, the behavior of seals can be a little different than in the strait. It's a very different environment. Animals up there tend to be, or outside of the strait, tend to be a little more skittish and so on. So worth testing whether or not these correction factors are relevant. So we were up there a couple of weeks ago for a 10-day trip, multi-purpose trip. But anyway, it took us a while to figure out how to actually capture seals up there with the kelp and current. But in the end, we were successful, and we have 5 deployments out. We caught 1 animal out at Pender and 4 at Wheaton. These are just the raw, unfiltered data, just to give you a sense. So we did both head mounted and shoulder mounted deployments. You notice we don't have that foam tag. We don't anticipate ever being able to recover these tags, just given the currents and so on, and also the fact that we're not there to immediately respond. So these animals, they'll shed the tag when they molt, sorry, and at the end of summer or early fall. And so the tag is simply shed off their pelage. But so to that effect, we have a ground station called a moat here, which is a listening station, and it'll log all the telemetry data from all the satellite tags that are within its range. And in the past, this has fleshed out our data hits by upwards of 30%. So we're actually, a team is going up there tomorrow, and they're going to set this up. We have permission to set this up, this ground station up on the cut block just above Telegraph Cove, and this will give us direct line of sight for our animals. And we're hoping they'll behave like they had, so far they have been, behave like our straight Georgia animals, where they just tend to really stay where we initially caught them. So yeah, just switching gears, our other project is estimating pinniped diet. So this is multi-species sampling. So all the sea lions, harbor seals. It's part of a larger project. So we're sampling pinniped for pinniped diets along key salmon migration routes and killer whale feeding grounds in and around Vancouver Island. So it's multi-area. So we are collecting scat for DNA meta-barcoding and also for hard part analysis to look at the species composition and the size of prey consumed. But there are limitations with scat, so we're also employing these other biochemical tracer methods, which are more integrative. Yeah, I'm almost done. Okay, to estimate diet. So we have a biopsy sampling program. We were up there in May. We will return in September, October to collect scats, as also to collect biopsies. So we have a crossbow biopsy system that we've developed to get biopsies from adult sea lions at this point, and we've been very successful in obtaining full blubber cores for fatty acid and stabilized isotope analysis. So I'll just wrap it up there and take any questions you might have. Thank you.
Awesome. Thank you so much. That's wonderful. Marika, it doesn't look, do we have any specific questions? I don't think so. I have a question.
Sure.
How do you catch the seals?
Oh, so so given the kelp, normally here we would kind of beach rush. We'd run in on a boat, and they usually stick here. They're not as skittish, and you can jump out and grab them with a hoop net. That simply doesn't work up there. So we've had to switch to our capture method. So we're tangle netting. So kind of like mist netting for seals, if you want to think of it that way. So yeah, we put a tangle net adjacent to haul out, keep our eyes on it, and come into the net, and we able to recover them and get them in the boat.
Fantastic. Thank you.
I have a question, if I may.
Yeah, absolutely, Jackie.
Hey, Strawn, how much do we get? What's the prize if we find one of the tags, the seal mold?
Wow.
Because there's people listening right now who are on it. It's great that they now know what they're looking at when they see a seal with a harness or a hat. But what's the prize, Strawn?
There's no prize. I don't know what people are going to do to get these things, but
no, drifting with the cap.
Oh, I know.
Not getting it off the seal.
So,
We're not those people.
Okay, let's just be clear. No, well, there's no float on them, so I don't know how long they'll drift and float. But wow, you want me to? I'll think about a prize. It'll be a big one.
I know some of the people on this call, I'm looking at the names and I think like James Wilson is going, challenge accepted.
Well, that would be incredible. And like to get all that additional data. Yeah.
I do know that Strawn isn't able to stay with us to the end, so I'll maybe just pause another moment in case anybody else has a question. You can type it into the Q&A. Or for those panelists who can't use the Q&A, you're welcome to just unmute yourself. All right. Okay. Wonderful. Well, thank you so much, and good luck with your with your seal hats this year.
Yeah, thank you very much.
We'll,
Yeah,
we'll see
much
you later.
appreciated.
Thanks.
Yeah, we look forward to me coming back up in the fall.
Yeah, wonderful. Okay, thanks so much. Terrific. So our next presenters are from CEDIS Research and Conservation. So I will pass it over to Emma.
Great, let me just share my screen. All right, can everybody hear me okay? And see my screen? Perfect. So my name's Emma. I'm the coordinator for Straight Watch Quadra, and I am here representing CEDIS Society today. So I'll start with just a bit about us. So CEDIS Research and Conservation Society is a nonprofit. We're dedicated to protecting cetaceans from physical and acoustic vessel disturbances. And we do that through education, monitoring, and then direct intervention, which I'll get into a little bit on the next slide. If everyone takes a look at this map over here, these are our locations around Vancouver Island. So we have Straight Watch South based out of Victoria, then we have Straight Watch Quadra Island, and then way up north, that's our warden program. So Straight Watch is one of our two programs. So Straight Watch is on water monitoring and data collection, and we collect data on vessel traffic, whale behavior, and then instant documentation as well. We also have a direct instant prevention component to our work too. So that's a very hands-on component, and that entails calling boats on the radio, letting them know that there's whales in the area. It means educating boaters on the water when there's whales around them, so we can directly educate those people who maybe don't have the information to watch whales safely and respectfully. And then it also means doing potential interceptions if there's a serious risk of a collision. We also have education and outreach components too. So we have events that we'll attend, community engagement, lecture nights, those kinds of things. This is just an exciting statistic that we got from our data last year. So we found that about 90% of boaters changed their behavior to be in compliance with the Be Whale-Wise guidelines after being contacted by us. And this is on the water only, so not including those outreach events. So this is after we've gone up to and talked to somebody, maybe after a follow-up of an incident or if we're doing prevention. So hopefully we see more stuff like this in the future. So if you're looking to contact us, that gray Zodiac up there, that's the boat that you'll see most often around Quadra Island. You might see it a couple times up north, but for the most part, it'll be down here. Our call sign is just Straight Watch on the radio. We're always monitoring channel 16 and 7 Alpha as well. I included the contact information for Straight Watch South as well. I know this is mostly North Island folks. This vessel down there, the black one with the white hull, that'll actually be starting the season up in our warden camp. So you might see the Straight Watch boat around until the beginning of July. So just so everyone's aware. And then we have our email and phone number as well, if anyone has any further inquiries for us. So this is our warden program. So we have a warden program, the Robson Bight Michael Bigg Ecological Reserve. And what's really cool about this program, it's actually been going on since 1991. And then CEDIS took over that contract in 2005. And the warden program is a little different from Straight Watch in the sense that all of our data, or the vast majority of data that we collect, is actually taken from a shore-based observation station, Eagle Eye. So that's a photo of our wardens, Steph and Astrid, collecting data from our warden site, from our Eagle Eye site, sorry. And that's located directly across from Robson Bight. And the data that we take includes vessel traffic, vessel behavior, whale behavior, as well as, of course, instant documentation, our on-water component, which is the actual wardens. So they're out kindly asking people to respect the boundaries of the bight, which is 1 mile offshore the Sitka estuary, as well as doing any instant prevention in a similar manner that Straight Watch does. If you're looking to contact them on the water, this orange Zodiac is called Warden 1. That will be the one that's most often out there patrolling the boundaries of the bight. We also have this larger red vessel, the Nimkish. Like I said before, that black vessel will be up there at the beginning of the season. And then into July, this one will be around more often. If you're calling us on the radio, you can just use Warden 1 across all of our vessels, just easier to remember and easy for us to listen to. If you're trying to contact our data station, you can just call us at Eagle Eye over the radio. Or if it's after hours, you want to get in touch with us at camp, you can use Boat Bay as well. We have shore-based stations at both Eagle Eye and our Boat Bay shore camp. Here's our email and our phone number. The Warden phone for the on-water component and the Eagle Eye phone are different numbers. So you can contact both if you'd like. And I think that's all I have to say. So if anyone has any questions, hope that was short and snappy.
Very short and snappy. Thank you guys so much. I feel like I must have put the fear in everyone. Everyone's like, so speedy. I'll maybe just pause and see if anyone has any questions for Emma and any, yeah. Emma, I'm wondering, I have a question for you. What is the most common sort of incident that you're observing among sort of recreational vessels and whales from Straight Watch Quadra?
Great. So that question is actually, it's actually the same across all of our programs. We consistently find that vessels going above 7 knots within 1 kilometer of whales is the most common incident by a landslide.
Right. Fantastic. Thank you. Any other questions for Emma? Oh, there's a question. I'll let Marika say it.
Yeah, Jackson's actually asking how the 90% statistic is developed.
Yeah, so every time we do a vessel contact, we have data that we collect on that contact regarding the type of vessel, the activity it was doing. And then we also have a box afterwards if it was a contact where we were trying to prevent an incident or an incident was occurring in that moment, if their behavior was changed or if they continued on violating the Be Whale Lives guidelines after we talked to them. So that's how we come up with that statistic. We'll circle yes or no if there was a positive behavior change after we talked to them.
And I think Allie has a question. She's got her hand up, so I'll let her ask.
Yeah. How do you kind of figure out what speedboats are going? Is it just visual? Do you become familiar of kind of what 7 knots looks like, or do you have like a kind of scale? Yeah, I was curious about that.
Yeah, great question. So whenever possible, we like to use AIS so we can be really precise. Sometimes most of the time it's very obvious, just based on our visual on the water. If a boat's moving really quickly as opposed to 7 knots, they'll generally be generating a big wake, maybe being noisy. So we try and use AIS when we can, but yeah, if we have to eyeball it, then we will.
Mm-hmm. Cool. Sweet. Thanks for sharing.
No worries.
Fantastic. Okay, thanks so much, Emma, and thanks so much for giving us a really clear visual of your boats and call signs and everything for this year, so everyone knows how to get a hold of you guys. Wonderful. Okay, next on our agenda is you, Allie. So come on back and and share your screen.
There we go. Does everything look okay? You can see everything? Or not everything, hopefully. Oh, sorry. Can you guys see everything?
Yeah, we can see it.
Yeah,
You look
great.
good. Yeah.
Great. Sorry. Awesome. Yeah. So I guess I'm not on the first slide. All right. Hi, everyone. My name is Allie, and I'm just so pleased to be here tonight. Thanks so much for putting this on. And it's such a pleasure to be part of the amazing work that's happening on the North Island. So I'm going to be chatting with you about my work looking at boater behavior around marine mammals in the Johnstone Strait region to help inform boater education. And here we have on the right, domino breaching last summer in the area. And so I just wanted to introduce myself a little bit more, share who this work is being done for and by. I have a background in marine biology, and this project is actually my current master's thesis right now. However, I've also spent a lot of time guiding, doing outdoor education and science communication, which I'm also really passionate about. And this ties in quite a lot with how I work with MERS and kind of the objectives of this project. And most of all, this project is being done for the whales, along with all of our work here and as well as for anyone who spends time on the water with marine mammals. So yes, this is largely for whale conservation, but also for boater safety and knowledge and how to coexist with these amazing animals safely. So this project is primarily partnered with MERS and their education campaign, along with the University of Victoria and my work there with the Applied Conservation Science Lab with Dr. Chris Darramont. So yeah, my work is aiming to provide a framework to better understand our interactions with marine mammals. We're focusing on interactions with humpbacks, but this project is aiming to kind of look holistically at the area. So we're including all marine mammal species as well as all boater demographics as well. So our main objective is to help inform and provide direction to the awesome MERS See a Blow Go Slow campaign. As many of you know, education and community awareness is just so critical to conservation and safety. And so amazing as this campaign already is, this research can help provide some direction to make this effort a little even more effective. And another goal of this work is to help inform future management decisions, to create some really helpful information regarding the regulations and guidelines in the region. So as probably many of you have seen, there's increasing vessel and increased humpback traffic. And this has led to a greater risk of vessel strikes, along with many other marine mammal species being at risk. And this has become more and more of a threat to both the mammals, but also to boater safety. So to try and help the threats of vessel collision to both whales and boaters, DFO has placed marine mammal guidelines and regulations, and they can be pretty confusing. So we're trying to kind of parse this out, and we also just don't have a great grasp on what's actually happening out on the water. And so this leads into my first research question, and this is to ask, how are people interacting with marine mammals in Northeast Vancouver Island? So we are looking at distances in regards to the regulations to have an idea of how they're being used and known out on the water. But we're also looking at other indicators of interactions, such as number of boats near a whale, how much time is being spent with a whale as well. And so digging a little deeper into this, the second question and aim we're looking at is, what are the traits of boaters and marine mammals that may explain the interactions between them? So, and by traits, I mean, for example, the general vessel types like cruise ships, commercial fishing, whale watching, or recreational fishing. And the species involved in the interaction, again, focusing on humpbacks, the whale's behavior at the time. We're looking and trying to identify the individual whale or clan, and if these interactions change depending on where they are in the region. So how are we actually going about this? This project is completely land-based, and we use a couple of different methods throughout Johnston Strait, Blackfish Sound, Queen Charlotte, and Waiton Pass. But yeah, the main way to see these interactions is when there is a whale in sight, we follow it for as long as possible, then make observations of any boats that are within 1 kilometer of this whale. So we understand that not everybody may see this individual, so that's why we're taking other variables down, such as kind of time, when the boat is spending within the distance of the whale. And this is also when we make note of the general vessel type, species, individual, like the individual whale or clan, its behavior, and whether it's alone, in a group, or with a calf. So we're going to be working throughout the region. And this leads us to our next steps for the project. I'm in the midst of planning the full field season this summer in July and August. And we're hoping to have a complete study and results by the summer of 2024. So thank you so much. And I'd love to answer any questions if you have them. And my call sign is Alley Cat on the radio.
Thanks, Allie. That's fantastic. Any questions for Allie? I'll pause maybe a quick minute if anyone has anything. Great. Okay. Awesome. Well, ticking right along then is Jackie.
Hi, everybody. Thank you so much for being here. and listening to the updates for the work being done in our area. I have the joy to be representing the Marine Education and Research team. I'm Jackie Hildering. We are based out of Port McNeill in the territory of the Kwakwa speaking people. The vessel that we are most often on is the Fluke, seen here. That is also the call sign, and we're most often operating under Marine Mammal License number 57. The work that we're doing is continuing population monitoring for humpback whales, and many of you have contributed to that work as well. Who are the whales? How are they using the area? Yeah, things like exciting news that Guardian's got her first calf, age of first calving and other life histories. Where are they? So supporting site fidelity, and indeed also opportunistically collecting fecal and prey samples. The cataloging, then, I would like to share with you that there will soon be an updated catalog. The holdup is me right now, but we will have an updated catalog for everybody from these population studies coming out very soon. So anybody who's paid for one in the past will automatically get an update. I thought I would also share with you some of the results, and so many people do share sightings with us, photos allowing for IDs, that this truly is, in addition to our work when we're in the field, so much is achieved through other people being in the field and sharing the information. So preliminary, emphasizing that, because we still have a backlog of a little bit of data, and things change over time as well when we figure out that, oh, that's who that whale was, and numbers increase between years as a result. But for now, with the analysis that we've done for 2022, in our core study area, as we reference it, there were 112 individual humpback whales, not sighted at the same time, but 112 different whales. In the Campbell River Comox area, there were 137, and there is overlap between these two, that some of the 137 were seen up in the core study area and back. And then outside of where we are discussing projects today, but wanting to share, if you haven't somehow picked up the well-covered media release, that last year was a record year for the number of individuals sighted within the Salish Sea. And that allows me to acknowledge as well that any information that we're collecting with regards to humpback whale IDs, where they are, etc., and what you are contributing feeds into the provincial effort. So DFO stopped cataloging individual humpbacks in 2010. The folks at the bottom here along different timelines, folks and organizations, had kept up the cataloging effort, and that is now under the umbrella of the Canadian Pacific Humpback Collaboration, of which MERS is the coordinator. So that feeds into anything coming in, feeds into the provincial effort, and it also feeds into efforts to understand humpbacks for the purposes of conservation across the whole Pacific. Otherwise, what we're doing is looking at why are the humpbacks feeding the way they are? Yeah, so looking at variables like what prey is it, what density of the prey, how many birds are involved, of what species. And here again, knowing that there are many people on this call who have helped with this as well, and certainly help with educational efforts around feeding strategies. We are sharing with you that where it was 2 individual humpbacks that we found with this novel feeding strategy, trap feeding, in 2011, it is now up to at least 30 that we have found that do this, again, under very specific conditions. So feeding studies continue when we're out there. What else are we doing if you see us out there, when you see us out there, is continuing the SCAR study. So looking at, by getting photos of the tailstock, and this is actually when we're likely to be the closest to the humpbacks, is to look for signs of wrapping most often around their tailstocks. So as you can see here with Argonauts, this whale that is so often in a predictable place up in around northeastern Vancouver Island, somewhere along the line, there's the evidence of a wrap scar here that shows that he is one amongst the many who have been entangled and have survived. And the importance of doing this scar study work is because humpback whales not organizing themselves like orca in families, if a humpback disappears, you can't know if they're dead or somewhere else. Hence, you need the evidence of looking for scars. And we've expanded that SCAR study work to, as mentioned by our colleagues at Oceanwise, to work in a collaboration where their expertise in looking from on high are collecting then imagery to let us see, are there scars that we are picking up on better from the air than we are from flush, and then comparing those two methodologies to see how that works. And if you're interested in the whole methodology, of course, I can answer your questions, but this is also part of the presentation we did from the Research Associates Night, so we can provide you with that link. And then otherwise, we continue to do systematic surveys to get baseline data to see the distribution of cetaceans throughout the year, if it's different over seasons. And when we're doing this work, we are often on the Merlin under contract and have the joy of working with Jared in that regard. So that is what we're doing. And thank you to so many of you who helped these efforts.
Great. Thank you so much, Jackie. Fantastic. I feel like we have a very quiet crowd tonight. Just going to pause for a second to see if anyone has any questions or needs any clarification. You're also welcome to ask questions about other presentations that you've seen if things have come up as well. Okay. Fantastic. Well, because we are ticking along quite quickly, which is fantastic, we will maybe take the opportunity to unmute everyone at the end too, and we can also ask questions verbally at that time if that also allows more conversation as well. So next on our agenda, I have to pull up, after Jackie is Sheila. So Sheila with Fisheries and Oceans Canada, I will pass it over to you.
Thank you very much, and thanks for the opportunity to present today. So I'd just like to, for those of you who don't know the Marine Mammal Conservation Physiology Program, just give you a brief overview of what we do. This program started in 2017, primarily involved with identifying anthropogenic or human-based impacts to the southern resident killer whales. And I'll just talk a little bit about what the program does and what our field season is going to entail. So we undertake research to increase our understanding of those impacts on all cetaceans. And right now we're really focused on southern residents. We do a fair bit of data collection on the northern residents because that population is doing quite well, increasing 2 to 3% per year, whereas the southern resident population is, depending on where you look at it, languishing and not doing very well, especially when we start looking at health parameters within that population. We can see that the southern residents compared to the northerns are just not as healthy, and we're trying to look at what the impacts are that relate to those health issues. We also are undertaking
sorry, I'm
research.
just going to interrupt that we can't see your slides.
Oh,
sorry.
I'm so sorry.
No, that's okay.
Do you see anything?
Just you, yeah.
Oh, all right. I've got it on share.
Oh, there we go.
Is that?
You're back.
Oh, good. I'm
we
so
go.
sorry. There you are. Okay. Well, the first slide was just that. So the second slide, just walking through some of the things that the Marine Mammal Conservation Physiology Program does. So we are currently working on humpback whales as well, looking at humpback whale habitat requirements, because as you all know, the humpback whale populations are significantly increasing. And what we want to draw attention to is how that fits into the ecosystem, and looking at the carrying capacity of the ecosystem and understanding a number of factors related to humpback whale population health and the ability of our ecosystem to support those significant increases. And of course, most of these studies are involved with species that have been identified as at risk, and we really work towards providing advice on ways and means to support survival and recovery of the species. So you're able to see the screen click. Okay. So as Jackie had indicated, we also fly the yellow pennant, which indicates our research license. Primarily, we are flying Marine Mammal License, or MML06 flag, which is the permit under my name, but occasionally we will switch to MML01, which is Tomás Daniel Velcroz's permit, if, for example, we encounter a species that's covered under his permit, and we, for example, North Pacific right whale or something along those lines will be flying his flag. We primarily use 2 research vessels, although we also work quite closely with Jared and the Merlin from time to time. The vessel on the top is the Great Northern. We usually have the pulpit on the front, so it's easy to identify. And the vessel below is the Charlie C, and that's a Zodiac 753. Both of them have research in white written on the tubes, and the names are across the radar arch on the back. So hail out and say hi. We're always happy to chat with people on the water. The Northern Resident Killer Whale Research will be doing at least 3 deployments this year. The first one leaves in a week, June 24th to July 10th, and that'll be out of Shearwater up in Central Coast. Then we move to Port Hardy, which will primarily be humpbacks, but potentially Northern Resident fecal samples if we encounter them. And then September, approximately the 5th to the 16th out of Alert Bay. The Northern Resident work is primarily collecting fecal samples in support of metabolomics and the microbiome research. This is a collaboration with Stanford University, and then looking at contaminants with Dr. Tanya Brown out of Fisheries and Oceans Canada. We also look at hormones, and this year we're going to be expanding that study to develop a health index. So we compare the northern population to the southerns. This is a volcano plot of fecal metabolites. And in the blue are metabolites that are significantly lower in the northern resident population than the southerns, and in the red, significantly higher. And this allows us to look at the differences between these populations and understand stress and gut function and health. So this year, rather than looking just at northern and southern resident populations, what we want to look at is individual body composition. So we'll be flying these DJI mini drones to get a body comp eye patch ratio with every fecal sample that we get, so we can start developing more of an individual health index. With Dr. Lance Barrett-Leonard, we've gone through all of the biopsies that have been collected, both through his efforts way back in his PhD and right through to current DFO efforts to try and catalog and bring that really valuable collection of data together. And we've run full genome analyses on these. There are, as we undertake this analysis, we can see gaps. So there might be occasions where we want to fill in some of those gaps with specific individuals. So occasionally we will be taking biopsies from northern residents. And we also take biopsies from the humpback whales. And I know Joanne's going to be talking about her work and what she does with those biopsies. Within my program, we're looking at lipid composition, comparing that to body morphometrics from photogrammetric images. And we're also undertaking a full genome analysis to look at specific differences, if they exist, between the northern and south, or between, sorry, the Mexico and Hawaiian individuals, and then those individuals that like to bounce between those two winter locations. And before I run off, I'd like to just point out that Fisheries and Oceans publishes something called Field Notes every year, and this describes all of the research that goes on in all the different First Nations territories. So this is a link to that document, but you can also Google it. So I'll leave it at that because Joanne's going to talk about what she's doing with humpback whale biopsies. Thanks very much.
Great. Thank you so much, Sheila. I had no idea about that Field Notes. That's a really handy resource. That's fantastic. I'll just pause again and see if anyone has any specific questions for Sheila. Sheila, I maybe have a question. Do you have a sort of goal number of animals that you're hoping to sample? Or is it sort of just opportunistic what you find?
For the humpbacks, you know, it's challenging. It really is opportunistic because you know, getting a fecal sample on command isn't something we've mastered quite yet. So we try and expand that. But because this year we have 5 years of fecal samples in the freezer, but this will be the first year that we're getting the alignment of the timing of photogrammetry with the fecal sample. We're going to be working with Lance and others to look at the closest time period that we can have a photogrammetric image, but this will be really nice to get those simultaneous images.
Yeah, wonderful. Thank you. All right. Okay. Well, thank you so much, Sheila. Jackie, I'm just seeing in our chat. We'll share that link again to the field notes into the chat, hopefully, so that everyone can see it, or we can make sure that it gets sent around for those of you like me that weren't aware that DFO had that great publication. So we'll make sure that that goes out to everyone as well. Oh, and look, there's a question too. Since we have some time, I think we can just ask those right now, Marika.
Yeah, the question from Donna is that since other studies are using fecal samples for DNA, why is it that the biopsy method is necessary for DNA?
Because we actually don't know which animal produced the fecal sample. So that's why we're actually trying to get that DNA library. When you get a fecal sample, you're following 5 or 6 animals and you don't actually know which animal it comes from. So it's really important for us to expand that genetic library so that we can identify which animal the fecal sample came from by running the DNA from the fecal sample. I've just put the link to the document in the chat as well.
Okay, perfect. Thank you so much, Sheila. That's wonderful.
You're welcome.
Okay, awesome. Thank you. Thanks so much, Sheila. Thanks for the question, Donna. Next up is Joanne Moreau, a graduate student working with Sheila and MERS as well. I know that Joanne is, I'll just give her a little precursor, in a small village in France right now. So first of all, she gets kudos for being the person who is up latest or earliest, as the case may be, to attend tonight. But that also there may be some challenges with her internet connection. So we will try and troubleshoot as best we can. Over to you, Joanne.
Okay, yes, thank you so much, Caitlin. As you said, it is 5 a.m. and I have poor internet connection, but we'll try my best. Can you guys see my slides okay? Yes, perfect, thank you, okay. Amazing. Yeah, so thank you so much for the opportunity of having me tonight to talk a little bit more about my research. I am a research associate with MERS, as well as an aquatic science technician with Sheila's program. Looking at well diet and recovery, mainly in the northern part of the Strait of Georgia. So yeah, as Sheila said, we various groups from Fisheries and Oceans, but mainly the MMCP program has collected humpback whale biopsies in 3 foraging hotspots, including Queen Charlotte Strait, the Strait of Georgia, and the Juan de Fuca Strait. But my research mainly focuses on the Strait of Georgia, more particularly that northern part. And it is a 2-part project. First, we use stable isotope analysis to determine diet composition from those biopsies taken. And then I will use an ecosystem modeling approach to predict the food web effects of the recovering humpback whale population on in the Strait of Georgia. But most importantly, what I wanted to talk to you about tonight is how exactly do we use a biopsy sample to determine a diet composition? Because this link between the two might be a little vague, and I know that Strawn with the Pinniped group has also touched on very briefly on that. So my hope is that to make you understand what we do with those biopsy samples. So what are stable isotopes? They are different versions of the same elements. In our case, we use carbon and nitrogen. And if you need a little refresher on those, I have to take you back to your high school sciences, a table of elements. But all you really need to know is that different elements not all elements are created equal. And while carbon will always be carbon, there are some heavier and some lighter versions of the same element. And therefore, these are called isotopes. And these biochemical markers accumulate in animal tissue. They just form everything in our bodies. And different ratios of these isotopes may create different signatures. So the theory is that each different species have a specific isotopic signature. And most importantly is that the isotopic signature is transferred from prey to predator throughout the food chain. So let's put this into context. Just hopefully really for you to understand the biochemistry behind all of this. So here we have our three of potential prey species with each of their isotopic signature reflected in that little colored circle. And our hope is that by analyzing the humpback whale skin and perform stable isotope analysis on this, we can see what proportion of each of the prey species make up that diet. And so note that these are hypothetical values, but this aligns with preliminary results that we've had so far. So it is still a work in progress. We have biopsies from last year and we'll complete with biopsies this year. Yeah, this is something that we hope to see and be able to determine the contribution of each prey item to the diet composition. And why does it matter in the end to know what humpback whale eat? Well, for us, it's very satisfying, not for a scientific community, know why the humpback whales come around Vancouver Island. It will also help us provide guidance of how much food is needed for the humpback whales, not only for the present population, but also for the future population. And this is where the ecosystem modeling comes in. But it will also contribute to the efforts of other groups, including MERS and many who have presented tonight to determine the ecological role of humpback whales around Vancouver Island. And with that, I hope you heard the entire presentation without saying I would to thank everyone who have been collaborating on this project from near or far. It's been a great pleasure to have such amazing collaborators, and I will take any questions.
Wonderful. Thank you so much, Joanne. We just lost you there at the very, very end, but we could hear you very well. So thank you, 5G hotspotting from Small Village in France. It was it held out for us all. I'm wondering if anyone has any questions for Joanne. So I'll maybe just pause. I have a question, Joanne. I'm wondering, I'm assuming, but is this the last year of field work that you're doing and you're writing up after this season, if I understand right?
Yes, for myself, it would be the last there could be a possibility of next summer, but I don't see that as feasible as I would be graduating, and see Sheila is giving me the no. But no, she wants me to graduate in time. But for this specific project, it would only be looking at the 2022 and the 2023 samples. And it is why we also have that between year gap to see if there's any variations, not to just get all of our samples from a single year.
Right.
But yeah, expect a paper in the next few bit.
Wonderful. Any other questions for Joanne? Okay, awesome. Thank you so much, Joanne. You're welcome to stay, but if you need to go sleep, we also understand that very, very much. Thanks for joining us from afar. All right, well, we are on to our final presentation of the night, Valeria and Lance from Rain Coast. And then once they're complete, we'll sort of open it up again and let anyone ask any other questions, or if anyone has anything else that they hope to share, we can do that at that time too. So over to you, Rain Coast.
All right, can you guys see not the notes, but the presentation?
Not in presenter mode yet.
About, is that working?
Yeah, that looks good.
All right, so I'm Valeria. I've been a co-director Rain Coast Cetacean Conservation Research Program with Lance since the end of 2021. And before going into the details of the program, a little bit more about Raincoast for those of you that are not familiar with Raincoast. We're an NGO that has existed for 23 years now. And we're basically a team of conservationists and scientists that work together to build support for decisions that protect the lands and waters and wildlife of coastal British Columbia. And we use peer-reviewed science majorly and community engagement to further conservation plans along the coast. And we call this approach informed advocacy. And so the Cetacean Conservation Research Program is a fairly new program at Raincoast. It investigates the biology, ecology, and behavior of whales, dolphins, and porpoises. And it focuses on how cetaceans use sound on killer whale health, nutrition, and genetics in the face of ecosystem change, and trends and impacts of underwater noise. And so LANS has been a presence on this coast for several decades now, but working on this coast is relatively new endeavor for me. So we thought we'd start this presentation by introducing myself and our team. So a little bit more about me. I started my career as a marine mammal research scientist in the 90s, actually, when I worked for John Leon and his whale research group in Newfoundland on both the humpback whale disentanglement program and on a wonderful humpback whale project that was called YONA. Some of you may have heard of it, the Years of the North Atlantic Humpback Whale Project, which was sort of like an international collaboration to study North Atlantic humpback whales across most of their known range. And then I got sidetracked studying canids for a few years, both red foxes and wolves for my master's. And in 2002, Lance took me in as a PhD student and never got rid of me. I studied beluga communication that took place at the Vancouver Aquarium and in Hudson Bay and the St. Lawrence Estuary. Then I became a research associate at the aquarium and worked very closely with Lance and the Marine Mammal Research Group there. And then became a full-time research scientist and was there through the initial transition of the Vancouver Aquarium to Ocean Wise. And through that time, I led beluga projects in the Arctic and Hudson Bay and the St. Lawrence, mostly related to beluga communication and impacts of noise. And at the end of 2021, both Lance and I moved over from Ocean Wise to Raincoast to start the wonderful cetacean research program at Raincoast. And our program grew very quickly. This is our team. We have a number of projects on the go, and we won't be talking about all of our projects today, but I'll briefly introduce our core team members. Adam Werner, because you will be seeing some of these faces this summer, I'm sure. Adam Werner is a conservation geneticist that has helped launch the Raincoast Conservation Genetics Lab. And he's doing really cool projects on killer whale genetics. Mariana is a staple and is a research technician for both our beluga projects and our killer whale projects. And she will not be with us on the water this year, but you'll see her with us on the water all of next summer. And several of you know Sharon Kay, who is working on her master's through the Raincoast Applied Conservation Science Lab, and will be with us in the field this summer on North Vancouver Island. And she's leading a great study on how social dynamics and prey sharing influence the health of northern resident killer whales. And then Maureen Gordon and Susie Hall, who may be familiar to some of you guys, have been doing a great job coordinating and implemented a very collaborative project called Noise Tracker. And so we will use the rest of the time to fill you in about our photogrammetry study, because that's the project that will have us all in the field most of the summer. And I will pass it over to Lance, who has been working with colleagues on photogrammetry for nearly a decade now. So over to you, Lance.
Hey, oops, thanks very much. Can you hear me?
Yes.
Yeah, that was a great intro, Valeria, but I think that's our 5 minutes. How are we doing, Caitlin? We've got 2 minutes left? Okay. Great. I'll just talk very briefly then about the what we're going to be doing in Johnson Strait in particular, up in your part of the world this summer. I will be working on the killer whale photogrammetry project again. I've done this for many years with OceanWise. I've moved to Raincoast recently, as Valerie mentioned, and we will be up there doing this work from August 15th to September 8th. And these pictures actually are supposed to say September 2015 and 2016. It's just an example of how quickly condition in killer whales can change. Next. Sorry, Valerie. Thank you. We'll be, as we've done in the past, and I began this work working with John Durbin and Holly Fehrenbach in 2014, but as in the past, we'll be measuring the length and the width and the shape of individuals from their photographs in order to determine their nutritional status, pregnancy rates, that sort of thing. We're going to be exploring the relationship between noise exposure and body condition. I'll talk about that more in a sec, and measuring growth rates in relation to prey abundance. Ultimately, we've always hoped that this information would be used to help the fisheries department make decisions about opening and closing fisheries in years, or closing fisheries specifically in years when there's very poor body condition and low Chinook catches at the same time. Next, please. And then there's the new component that we'll be working on this year, thanks to some funding from DFO. Thank you very much, DFO. The title of the project is The Relationship Between Body Condition, Fecundity, and Mortality in Northern Resident Killer Whales. And we'll be comparing body condition of northern resident killer whales in areas where there's relatively high traffic, i.e. Johnson Strait, eastern Queen Charlotte Strait, to the body condition of whales up on the Central Coast, shown up here on the top left of the map. That's the Central Coast from Millbank Sound to Camano Sound. And so we're excited about that. I've worked, as I said before, I've worked with OceanWise on this project for many years, with Brittany and Gary in particular. So we'll still be collaborating behind the scenes on data analysis, but we'll be working independently in the field. And the last slide. And so you'll see us, Valerie and I, on the water this summer in Johnson Strait in August on this boat. It'll be familiar to many of you. It's the Achiever, the Raincoast Conservation Foundation's longtime research platform. It's spent a lot of time in the area in the past, and it'll be equipped with a platform actually, instead of the boom and the mainmast, to fly the drone from, and we'll be also working from the Zodiac. And our research permit number is MML18. So you'll see us on the water at that time. Looking forward to seeing you guys. And that's about it for me.
Thanks. That was perfect. Thank you guys so much. We are spectacularly 20 minutes ahead of time, so we have lots of time to chat or ask questions. So Marika has her hand up. It looks like we have a question. Go ahead, Marika.
Lance, based on how you're measuring sound impact on whales that presumably are moving between quiet areas and not so quiet areas. So is it a snapshot in time, or is it based upon how much time they spend in those different zones?
Yeah, it's good. You ask good questions, Marika. You always have. No, so what we're going to be doing is comparing the body condition of northern residents that spend most of their time summering in the Johnson Strait area. So, you know who those pods are, mostly A-clan whales in the I-15s, and to the body condition of whales that spend most of their time summering on the central BC coast, and those would be mostly R-clan and G-clan whales, not exclusively by any means. And that's the way in which we will be addressing that issue of whales moving from one area to the other. It's an imperfect science, of course, because the whales carry some condition forward with into the summer from the previous winter. But by the end of the summer, we've certainly seen in the work that I've been doing and involved with and collaborating with OceanWise and NOAA on over the years, we can certainly see a difference in the southern residents, at least, between May and September. So we think that by late summer, they will have the body condition will to a considerable extent reflect how much they've been eating in the last few months. when they've been separated in the way that I've mentioned.
Wonderful. Thank you so much, Lance. Marika's just unmuting you all. She's adding you to the chat. Anyone can raise their hand or unmute themselves and ask questions.
Oh no, here we go.
And maybe while you are pondering that, I have a question as well. Lance, I know for many years we've talked about how the hope is eventually that sort of photogrammetry monitoring will allow us to potentially do more fine scale fisheries closures or openings or so on and so forth. I'm wondering how close you think that is.
Yeah, it's a good question. I think it's I don't really know how close it is. It's not I don't have any power to, of course, I can be persuasive. I can sit on the sidelines and sort of say that I think this is important. But this is really the kind of decision that fisheries management has to make. But I think there's I mean, we've seen DFO being very responsive to data that we worked up. We, in this case, working closely with the folks at NOAA, showing that really being a big part of the evidence that southern resident killer whales are food limited, are prey limited, at least in some years, not necessarily in every year. And I maybe I flatter myself, but I think that that's part of the reason that DFO has has really pulled out the stops to restrict fishing at certain times of year, to reduce the certainly local competition in the Salish Sea for Chinook between killer whales and southern residents and fishermen. So I don't see any big obstacles myself to this, this sort of an index of body condition combined with Chinook abundance being used as a sort of an in-season emergency tool, perhaps, to raise the warning flag that in-season closures or restrictions in Chinook harvest are going to be necessary. But really, we've still got a bit of work to do, I think, to make that systematic, to be able to provide the very kind of convincing systematic evidence to fisheries managers sort of in real time. So that's a goal. I think we're getting close to it, but we're not there yet.
So I got a question for you, Lance, from Donna, who's asking how you are measuring noise exposure.
Well, we're not going to be measuring noise exposure directly. We're going to be inferring. We're not going to be putting D tags on the whales or measuring it in that kind of direct way. But we'll be inferring. We know that we will be quantifying vessel traffic to the best of our ability. So we'll be able to say that. We already have good evidence, of course, that's why we've got the experimental design that we do, but we'll be able to quantify vessel traffic moving through the areas that the whales are, these two different study areas, and we'll use that as a proxy for sort of noise and disturbance, I guess.
Fantastic. All right. Any other questions? We've still got all our panelists except for Strawn, who had to leave us, and lots of folks who are going to be out on the water themselves. So yeah, happy to answer any other questions, or if any of the panelists had anything else that they wanted to add or ask of each other, you're welcome to do that as well right now. Oh, I've got some raised hands. Go ahead, Marika, you can. Oh, you're muted.
Go ahead, Sharon. I'm going to unmute you.
Still not working out with that mute. I just had a question for kind of everyone on the call, presenters and anyone who's listening. If anyone has any kind of requests about real-time field season updates on what we're seeing out there on the water. And if people have any platforms that work really well for them, so social media, you know, sort of field notes at the end of the season, blogs, just wondering if there's an opportunity here for people to sort of speak about what is the best communication that people find for getting real-time research updates, if anyone has any suggestions.
Thank you.
If not, you can always email us.
Thank you, Sharon. It wouldn't surprise me if like you may have access to the PWWA app that you know exists. There's a lot of cases, like I think per species subsection of the community, it depends on, like we have different ways of communicating. So if it's humpbacks for Northeastern Vancouver Island, for certain groups, we have a running list of which humpbacks are already in the area. There's the feedback we're providing directly to data contributors, to give that as an example, where that is like person to person or person to group.
Right, yeah, Jackie, yeah, and just kind of a reach out to the whole community if anyone has any other little preferences, just so that we know how to best communicate results in real time. That's awesome.
I think in terms of like when boats are in the area, something that is really valuable is to, via NIMMSA and ourselves maybe, where we can distribute it to people who are on this mailing list, that we can let people know the reminder that, hey, you're going to see the achiever in the area. This is what they're going to do. Forwarding whatever information you have. We've done that, like for the Pinniped team, for example. But if it went to NIMMSA and us, there might be some cross posting, but it would get to everybody who's on, who was invited to this meeting.
Maybe I'll just add as well, Sharon, that one of the things we did last year, which isn't in season, but we did do a little post-season meetup as well, both in Port McNeill and Campbell River. NIMMSA hosted those, and it was sort of a great way to kind of check in sort of quicker time about what happened that field season. So you'll probably see invites for those. We did them in October in Port McNeill, like right after kind of the main season, and then adjacent to, I believe it was the NIMMSA meeting, right, Steph?
Yeah.
Which was held in, I guess it wasn't until December.
Yeah.
Yeah. Yeah. Great.
Got hands up all over the place. Lance, was yours a response to Sharon's question?
Oh yeah, I was just wanted to say I sort of feel I'm being conspicuously silent and saying talking about research updates. It's just with the photogrammetry work, it takes a long time to, you know, as Gary and Brittany can attest, you know, generally it takes a good chunk of the winter before you really can say much about the sort of overall condition of the whales. We can, if they're in really bad shape, we can see that pretty quickly, but the fine scale measurements take some time. So we're really happy to provide updates about what we're doing and kind of what our success rate for having encounters and that sort of thing is, and we'd likely do that in direct communication in Twitter and or at least on Instagram anyway. But for those detailed findings, it takes a little while.
And I'll just add for DFO's humpback whale data, we do upload all our sightings to Happy Whale. So that's publicly available. It's may not be real time. You don't always get them out there right away, but certainly from the season, we'll go through and put all the photos up. So all those sightings with locations are available. But for the biochemical analyses, obviously that takes a bit longer for us to slog our way through all of that. So thanks.
And Emma, was yours in response as well? Otherwise, I'll move on to Michael.
It wasn't. It's a separate thing.
Okay. In that case, Michael, did you want to ask your question, and then we'll go with Emma?
Sure. Regarding the entanglements, I know Jackie was talking about the SCAR studies, and I'm just wondering if you're seeing more entanglements? Is there a trend, and are any of them happening in the Broughton area?
Caitlin, T-motion wise, jump in when need be. So Michael, we don't know enough. So we are catching up. Like we will be able to, between the two methodologies, both at boat level and through the aerial methodology, we'll be able to start seeing acquisition of scarring upon scarring with enough time. Certainly we know of individuals that have been entangled in the Broughton. I think it's fair to say that humpbacks are likely to be entangled in all along our coast. But as far as a trend goes, we're not there because what is happening right now is sampling individuals, and some of the MERS data, again, not aerial, goes back quite a while. So we do already have layers of data looking at, okay, now there's an additional wrap scar around whichever whale's tail, but we just don't have the breadth of data yet. But as we acquire it through the two methodologies, we will be able to do things like look at where is this whale most often, where is it migrating from, and frankly, there's even not ideal protocols yet in when a known whale is disentangled to even know who that whale is and to have a system of place to know that there are resightings. And that's something as the Canadian Pacific Humpback Collaboration that we're working on with DFO.
Okay, thank you very much.
Sheila, I saw your hand go up. Is your response into Michael's question?
Yes, well, it's actually sort of, yes. And to Jackie, just to let you know that we are going in next week to do sat tag deployment training, and we will have sat tags on our boats. So if we do encounter any entangled whales, there will be a couple of more vessels on the water who can deploy those sat tags for disentanglement. So that's a real positive movement forward. And as we've discussed, we'll be really happy to provide any of our aerial data for the scar analysis as well, because I think we have at least 40 individuals from last season, and we'll be getting more this season. So happy to help out on that study as well.
Yeah, and I'm going to jump in to make sure that everybody knows what the relevance of the sat tags are, because not everybody may. So overlap with humpbacks and fishing gear along our coast, it's happened very suddenly that there's population growth and a shift from somewhere else. And therefore there are more entanglements. Again, through the SCAR study, we're trying to, with our collaborators, get a sense of how often that's happening. And there are more people being trained to put on sat tags, because most often it is not an immediate risk of death with humpbacks, but rather that the greatest limiting factor is finding them back. So these are tags that are put onto whatever the whale is trailing behind it, then set off a signal so that the expertise and equipment can come in to attempt the disentanglement, and it's the best chance of a successful rescue effort. So it's great that, yeah, that's ideally all research boats, guardians, and fisheries officers would know how to put a tag onto a whale, which also requires knowing what normal behavior is of a humpback to be able to be effective in that regard.
And I will let Marika continue to deal with the hands up. I thought I just mentioned, Michael, the second part of Michael's question was about whether or not entanglement is happening in the Broughtons, and certainly it is. There is entanglement that occurs with different fisheries, including most recently the one in January, I believe, right, Jackie?
Yeah, and it's so difficult to know. The one that was here, Michael, in like Wells Pass, the gear appears to have been from the Salish Sea. So there's that too, right? You can spot an entangled whale in an area, but again, to the point of it's so important to know what gear is it to determine the origin of the gear to thereby try to stop the problem at the source.
Okay, I think we're going Emma and then Gary. I saw your hand go up briefly.
Yeah, sorry, I just had a quick question regarding training for deploying those sat tags, and if if maybe Sheila or anybody else has any insight into how we could go about getting that. I'd spoken to Paul a few times about it, but I've not heard anything back since. So I'm just curious if anyone has any information on that, because I'd love to get some of that training.
That would all be through Paul, I would assume. And the sat tags aren't cheap. So that's some of the things. We've purchased 2 units this year, but certainly I would really like to see that program expand. So just keep at Paul. See if you can.
And Gary, what may be helpful is to reach out to what other researchers and those often on the water, like Straight Watch, may also not have training, so that there could be a group put forward to make that effective, or all the more effective.
I'll just jump in quick, Jackie. Straight Watch actually got some training done about a week and a half ago in regards to our sat tag training. So we have a sat tag that's now based on Quadra Island with several individuals who are certified to use it.
Great.
Thank you.
Am I all good to kind of proceed with my question now, unless anyone else has any other comments about that?
Yeah, I was just going to make sure that I said this because Brittany made sure to put this in the chat that the aerial images that are currently being analyzed by MERS may develop into showing some trends around entanglement as that data set gets worked through. Carry on.
No worries. No, that was an interesting conversation. I just had a question regarding the humpback abundance that we're seeing this time this year. And I'm not sure if this is just that what we're seeing down here, but we've seemed to notice that there's far more humpbacks this time of year than there has been last year and the year before that. We were on the water today and there was easily a dozen just between like Quadra and Middle Natch area down towards Savory. I was wondering if you guys at MERS or anyone else had kind of noticed that, or if that's just a kind of a micro trend that's happening around Quadra.
You're the ones who'll know, right? You're the local, most consistent, like together with the whale watching community down there. You will know, like this is only 1 year, but if there has been consistent efforts by CETUS over the same area, sorry, Emma, you know this, but saying it just for clarity's sake, you're the one who would know if this is something that is a market trend or not, but also like so valuable that through you, we know who the individuals are, because who they are, and then coupled again with like body condition is going to provide a picture of what's going on there.
Great. Yeah. Thank you, Jackie.
Go ahead, Marika.
Oh, never mind. Lance's hand just went up, so that solves my.
Well, this is just more of a comment than a question, I'm afraid. But I think this is really interesting about humpbacks in the northern Strait of Georgia and in Johnson Strait too. And I just, there's not a lot of us old farts on the call today, but I just want to say it's just a stunning change for, if you haven't been around very long. For those of us who have been, humpbacks were scarce, 20 years ago in the area, and this explosion of use of the area by humpbacks is just one of the most remarkable things I've seen in my career, I guess. And I think we, hats off to MERS and to CETUS and to everyone for documenting this so well. It's a great, this kind of recovery of humpbacks has been seen in other parts of the world as well. But I can't think of a place where it's been, perhaps the US East Coast, but there aren't many places where it's been documented as well as it has been here. And this incremental sort of repopulating of the coast in the summer, sort of showing up in Johnson Strait and having humpbacks around consistently. For many years, northern Strait of Georgia was a desert, a whale desert. We ran up in the boat every year. We never saw a whale out there. And then suddenly, 10 years ago, Jackie, 12 years ago, 11, I don't know. it got popular with humpbacks, and now you can walk across them, right?
Jesus. No, you can't.
Yeah, you can if you're wearing soft-soled shoes. I think isn't that okay, Sheila? I think that's in the whale guidelines. No, maybe not.
Dear God, people. Yeah, no. Yes,
you know what I mean.
humpbacks are back. Yes, I do know what you mean. And it is that it started, like, as you know, because you've been traveling up and down the coast doing killer whale work or in other roles in your life, that it started more in the north and has proceeded downwards. So like now what's happening, and thank goodness for the community of whale watchers in the, like generally, those who are contributing data, but between Straight Watch Quadra and dedicated members down there, thankfully there is more consistent effort than there used to be, where then exactly what Emma's asking, like if the starting time of Straight Watch is the same, there's the same amount of effort, the same amount of expertise, Emma, right, in knowing who the whales are and the dedication to delivering that data, then there's pieces of the puzzle that are being delivered that are really helpful.
I think it would be interesting to know, though, and I know there's lots of people on the call right now, but if other areas outside of where Emma is around Quadra are also seeing a lot more early season humpbacks, and I think maybe that was part of what she was asking there, is outside of that area, is it being seen, you know, maybe further down in the Strait of Georgia or up in Johnson Strait area, as well as more early season humpbacks this year. I think for Johnson Strait, it's pretty standard, but you would know better than me, Jackie. Yeah.
Yes.
And I'm not sure if there's anyone that can speak to maybe the area around Nanaimo or further down into the Strait of Georgia, but.
And with there, it's difficult to know too, right? Because the variability of the whale watching and when they're starting. But yeah, Tasley would be the best source for that.
Well, and the BC Station Sightings Network has pretty good coverage down south. But.
Yeah, as far as individuals go is what I'm saying, right? So Tasley would be the one that would have the finger at the pulse of that.
Fantastic. Well, I don't see any more hands up, so I will maybe do a last call for questions or any other comments that anyone or anything else anyone would like to share. Welcome to do so right now. Wait a moment. Great. Well, if there aren't any questions right now, I just want to thank everyone for joining us tonight. It was really wonderful to hear about all the projects that will be happening. It's really great to see over 35 of us, and I know many people were multiple people watching at once as well. Kind of just learning about what's happening on the water and continuing to really act as a community around Northern Vancouver Island, supporting each other, understanding what we're doing, but also being able to see each other's faces. And so we know who is out there. So I'm wishing everyone a really wonderful season. We hope that we will see you all in October or December for sort of those season wrap ups so we can talk all about what happened in the year. But please reach out to one another if there are further questions. We did record this, so if you have colleagues that weren't able to join us today, we will be able to share that. And if you need to get in touch with anyone and you didn't happen to get their contact information, you can always connect with us at MERS, info@merssociety.org, and we would be happy to make those connections. So thanks everyone for a great night, and we will hopefully see you on the water this summer. Thanks everyone.
Bye-bye.
Thank you.
Thank you.